I'm Jaren Citak. I am a undergraduate student at Mount Holyoke College. I work in the Woodard Lab doing Drosophila melanogaster genetics. Before coming to the National Institute of Basic Biology at the National Institute of Natural Sciences here in Okazaki, Japan, I had very little lab skills. I got a lot of my degree online during COVID and did a lot of virtual experience, but not a lot of hands-on. But here, it's been all hands-on all day long with some reading on the side, but a lot of micro-injecting, pipetting, doing PCR, doing microbiology, making plasmids, primer design, all types of things that I had no idea how to do a few months ago. I've also worked with a lot of Japanese people here in the lab, and they've taught me cultural customs alongside the science that I've been learning. Like after a long day of Sanger sequencing, we'll have a tea ceremony in the lounge. My lab back at Mount Holyoke College in South Hadley, Massachusetts, does a very different kind of genetics on Drosophila. We look at hormone signaling pathways, whereas here we're working on hard genome editing, which has introduced a lot of new skills to me that I think I'm going to implement into my research in hormone signaling, especially ecdysone signaling with the Drosophila flies I keep at my home lab. During this experience, I've been able to try a lot of new different procedures and protocols for different kinds of experiments I've never done before, some of which I had never even heard of. And it's been amazing to see them actually work. After learning how to microinject from the bare bottom and working my skills up, being able to look and take microscopy photos of my embryos that I actually was able to make successfully glow was incredibly gratifying. It was a really cool science moment and made me just want to do more experiments. This is a really cool and unique experience. It's awesome for a STEM person to actually be able to go and do this kind of cultural exchange, and it's been really rewarding to do a long day of working and learning all these scientific techniques and then go out in the world and experience a new country and culture that's completely different from what I come from. If you've gotten accepted into this program, I would say do your pre-research. Read lots of papers that are relevant to the research you're doing, understand the kinds of experiments and protocols you're going to be doing and why you're doing them, what do those certain reagents do in that reaction. I think the more you can inform yourself before coming here, the better. Not only for the scientific journey, but language skills, cultural skills, understanding of how things work here, super important. I did a ton of research before coming here and I still wish I did more. You could not be too prepared to be here. I work under Dr. Kiyoshi Naruse, working on Medaka genetics and genomics. Having Dr. Naruse as a mentor has been really cool because he's been doing this for forever, and so he's showing us the old techniques, the new techniques for all different kinds of things, all one after the other. So we're learning a lot of different ways to accomplish the same goals, like plasmid infusion and other ways to make plasmids that we need. Also comparing CRISPR-Cas9 to CRISPR-Cas12a and comparing the different PAM sequences and why those two tools can be used differently, and making sure that we know how to use both of them so that we can really decide what's more effective in the situation that we choose to use them in. This is the fish room. This is where we keep all different kinds of Medaka. Here there's all different kinds of genetically modified ones, like ones that glow in the dark, ones that have different colors, ones with tumors, no eye color, and curvy spines. These are our fish that we maintain. We feed them, we clean their tanks, we change their filters, we change their water. These are the babies. They're all grown up. Hi, welcome to the lab. Let's take a look. This is where we spend most of our time. This is my bench. This is one of my mini lab notebooks. These are our pipettes, some things. This is where I do my micro-injections using this apparatus here and this microscope over here. This is where we make our capillary tubes for micro-injections. We do a lot of those. These are the thermocyclers. Sometimes we'll use many of them at the same time to do different PCRs or other experiments. Let's look at our centrifuge. We have a few. Here's a centrifuge. Here's another one. This is the one we use the most. It's a bit finicky. Here's an autoclave. Over here is where we do lots of microbiology experiments with building plasmids with E. coli. And this is where we run our gels most of the time using agarose gel electrophoresis.